Hi, my name is Lexi Calloway. I'm going to be talking about the anatomy and enzymology of the digestive system, and then also fat absorption, or fat digestion. So let's start with GI anatomy. The GI tract ranges from the mouth to the anus, and when food enters the oral cavity, it goes from the mouth to the esophagus to the stomach, to the small intestine and the large intestine, and then out through the anus. Throughout this interconnected tube is where digestion happens. So this is the chemical and mechanical breakdown of food. And then the tube consists, or it's lined with secretory cells. And each part of the tube actually has kind of a different anatomical makeup, just depending on its function. But you can see that here in this picture. And even though that they are different in each portion, they do have similarities. So starting from the inside, or the internal layer, we have the mucosa, which is the inner lining of epithelium, connective tissue, and smooth muscle. We have the submucosa, which is the layer of areolar connective tissue that houses blood vessels and lymph vessels, submucosal plexus, and types of glands. We have the muscularis, which is the inner circular and outer longitudinal layers of the smooth muscle. And we also have the serosa, which is the areolar connective tissue and simple squamous epithelium, and that's the outermost covering. We'll do some just really simple GI organ anatomy. So we'll start with the oral cavity. There are glands that secrete saliva, and there's three of them. There's the parotid gland, the submandibular gland, and then the sublingual gland. And then there's also the tongue and teeth that help with the digestion process at this point. It goes into the esophagus, which is this tube, before it hits the stomach. And here there is the fundus, which is kind of this top curvature portion. There is the body, which is this middle open portion, and then the pylorus is the bottom curve. And then separating the stomach from the beginning of the small intestine would be the pyloric sphincter. So then once it passes through that, it goes into the small intestine and into the duodenum. And the small intestine is this brain-like looking structure in the middle here. So the duodenum is over here. The jejunum would be the next portion of the tube, and then the ileum is at the end. And then the ileum connects to the large intestine, which has an ascending, transverse, and descending limbs. And then it would travel out through the rectum. So this picture here does look a little bit overwhelming, so I will just kind of sum it up for you. We have the oral cavity, which secretes saliva, and there's enzymes in there that, along with the movement of the tongue and the chewing, that start the digestive process. And then within the stomach, and I know this says HLC, but it's supposed to be HCl. So HCl is secreted from the stomach, and what that does is it activates peptidogen into the enzyme pepsin. And then we have pepsidogen, which breaks down bonds that link amino acids. We have gastric lipase that breaks down fat, and histamine that induces the production of acid within the stomach. Then we go to the small intestine, which secretes CCK, or cholecystokinin, and that is actually the enzyme that makes you feel full, but it stimulates the gallbladder to contract and stimulates the pancreas to secrete enzymes. Then we have secretin, which stimulates HCO3 secretion and inhibits other acid secretions. And then we have GIP, and what that does is it stimulates the insulin release, and then we have GLP-1 from the large intestine. So now we'll talk about fat absorption. So you wouldn't necessarily consider, well, not knowing about digestion, you wouldn't consider the pancreas or the liver to be part of that, but they secrete enzymes that aid in digestion, so we'll talk about that in a second. But the digestion of fat is kind of complicated by the fact that lipids aren't soluble in water. So what the liver does is it secretes these bile salts that attach to the lipid droplet, like right around here. And the bile salts are actually amphipathic, which means that they have a hydrophobic side and a hydrophilic side. And the hydrophobic regions of bile salts are associated with the surface of the lipid, so that the polar chains can interact with, associate with the lipid, the surface of the lipid, so that the hydrophilic portion can associate with the water on the that will be surrounding it, so that it can move through water and things. And then that process actually kind of complicates the digestion a little bit further, just because two enzymes have to be released instead of one that could potentially, if it didn't have to do that. So that would be the pancreas secretes lipase and colipase. And what lipase does is it removes two fatty acids from each triglyceride molecule, which results in one monoglyceride and two fatty acids. And then for that to happen, the lipase has to travel through to the lipid droplet, but it's unable to because of the emulsion process of the salts around it. So then colipase has to be released so that it can kind of dissociate a couple of those bile salts, and so that the lipase can enter the actual lipid droplet. So after the emulsion process, and I don't know that I'm saying this right, but I'm going to say micellase. Well, a micellase is formed, which is the combination of fatty acids, bile salts, mono and diglycerides, phospholipids, and cholesterol. And what they do is they kind of travel in unison toward the enterocyte, which is one of the cells, which is the cell type in the small intestine that does the fat or continues the fat digestion. So the micellase then enters, so it travels toward the cell in this condensed kind of sphere. So this is a summary of the entire process of lipid digestion. So I'll just kind of go through what we've been through. There's the lipid droplets with the bile salts that do the emulsion process. They form micellase. They travel toward the enterocyte surface, and not all of the components of the micellase can be transported into the cell in the same way, so it kind of has to separate once it gets there. So the fatty acids and monoglycerides are absorbed primarily by simple diffusion, but cholesterol is unable to be transported in that way. So what happens is it goes across the brush border of the membrane, and it has to be transported with an energy-dependent, pretty much co-transporter. So then what happens once all of that is inside the cell, the monoglycerides and fatty acids enter the enterocyte, and then they move to the smooth endoplasmic reticulum, which is this part right here, and they combine to form triglycerides. And then the triglycerides actually combine with proteins and then again with the cholesterol, and they form this chylomicron. And then because of the size of the chylomicron, it can't be transported out of the cell. So it has to go within the Golgi apparatus and be packaged and whatnot, and then it will be transported. And it will leave the cell through exocytosis. And then the fat actually, or this chylomicron actually goes through the lymphatic system. So it's taken up by the lacteals, which are the lymphatic vessels. And then they pass through the lymphatic system until they enter the venous blood system right before it goes through the heart. So that is my spiel on GI tract anatomy and the fat absorption. Hope you enjoyed. Thanks.